Click for IB Biology. Here we're taking a look at what is called the Bacteriophage Lambda. A blank PDF of this presentation is provided in the description so that you can annotate your own notes as we go along. There's a quiz so that you can test your understanding of this part of the syllabus. I've provided a prompt structure so that you can begin your own research into greater detail. And then on the end screen of this video, you will find links to other videos in the virus sequence and a link to the channel and the next video. Bacteriophage lambda is an obligate parasite of bacterial cells, such as Escherichia coli, E. coli. So it's not just the cells of plants or animals that can be infected by a virus, but also bacterial cells. The Bacteriophage lambda can involve itself in both a lytic or the lysogenic cycle. And indeed, it would be worth using the Bacteriophage lambda as an example of a lysogenic cycle as you prepare for exam. The bacteriophage has a number of components to its structure. These include the capsid head structure here, which would contain the nucleic acid. There would be this region here, which is referred to the helical sheath, or the collar region, sometimes known as the collar region. And then there's a base plate with pins, so-called pins in it, which allow it to penetrate the cell membrane. And then there are these contractile proteins that look like legs. They're actually contractile proteins, and they are able to bring the Bacteriophage lambda into contact with the cell membrane. The bacteriophage has a preference for maltose membrane porins. That's like a pore structure, and they tend to lock onto those in preference to other locations. And having locked onto those locations, the leg structures pull the base plate down into strong contact with the cell membrane, penetrating the membrane, allowing the introduction to the double-stranded DNA, which is characteristic of the bacteriophage. Now, of course, the next part depends on whether or not we are going to go lytic or lysogenic. But were it to be lytic, then the bacteriophage will hijack the protein synthesis and DNA replication mechanisms of the bacterial cell, replicate itself, create these capsid phage structures before producing proteases to disrupt the membrane, to burst out, and then move on to infect other E. coli bacterial cells. Alternatively, it could be that the DNA here will incorporate itself into the host nucleoid. Now, in the case of bacteria and bacteriophage, rather than being called a provirus is in a eukaryotic lysogenic cycle. But in the case of the bacteriophage and a bacterial cell, this is called a prophage. And so once this piece of DNA here becomes integrated by integrase, of course, into the nucleoid, it is now known as a prophage. It would remain dormant there during the rapid cell division of E. coli, and in the process of doing so, it is also replicated before later entering the lytic cycle. Here's an image, a micrograph showing the structure of the bacteriophage. There are many different types of bacteriophage. This one, the lambda, the parasite, obligate parasite of E. coli, is specified on your syllabus. Appearing on the screen are links to the channel, to the next video and to the playlist on viruses.